Hi, I'm Mark Gormley. I'm a consultant in oral surgery and a welcome PhD fellow at Bristol. And I'm going to talk to you today about using germline genetics to investigate potentially modifiable risk factors in head and neck cancer. So as all of you here know, around 90% of head and neck cancers are squamous cell carcinomas that originate from the epithelial lining of the oral cavity, oropharynx, hypopharynx, and larynx. And we stratify based on subsite, based on potential differences in their etiology. However, they are primarily driven by smoking and alcohol, and of course HPV, which is linked with oropharyngeal cancer, thought to be sexually transmitted. But these three major risk factors are actually quite well correlated and difficult. It is difficult to unpick independent effects, and some of our work has attempted to do that. We also know, as we've discussed during the last few sessions, that the incidence of head and neck cancer is increasing despite a rapid decline in particularly smoking rates within developed countries, and therefore other less established risk factors which are known to affect other cancers like obesity and related metabolic traits also warrant some investigation. During my PhD, I used a number of different approaches, but for this talk, I will focus mainly on Mendelian randomization, which is a genetic epidemiological technique, and it uses large population data sets. And we're very fortunate at Bristol to host Head and Neck 5000, which feeds into the Game On network, of which many of the Headspace and Voyager studies are part of. And we also have a number of cases in UK Biobank in which we can replicate some of our analyses as well. And as Tom actually talked about during his talk, in order to look at more downstream causal effects using techniques like Mendelian randomization, we need large genome-wide association studies. And we can use these population data sets to do that, where we have both cases and controls genotyped using SNP-chip array panels. We can perform statistical associations in order to identify SNPs or genetic variants which pass multiple correction and QC, and these will be statistically associated with a trait, for example, smoking, and we can use them as instruments in frameworks such as Mendelian randomization, which I'll talk about on the next slide. I also tried to follow up some of my findings using experimental assays where indicated. So for any of you who've been unfortunate enough to sit with us over lunch, we've probably made an attempt to try and explain Mendelian randomization. And it's analogous to nature's own randomized control trial, whereby during meiosis, you get the independent and sort of random segregation of alleles. And some people will inherit alleles or genetic variants which may confer a protective or risk effect. For example, you may actually get lower cholesterol, lifelong lower cholesterol, whereas other people may inherit a wild type allele and have more normal levels or even higher levels of cholesterol. And you can follow these people up to a disease outcome, for example, cardiovascular disease, or in this case, head and neck cancer. And it's a powerful tool to attempt to overcome some of the limitations of conventional epidemiology, such as confounding, and in particular, reverse causality, because we know that the genotype always precedes the phenotype. However, like any scientific method, there are some assumptions which, if violated, could lead to misleading results. And the first one of those is that the genetic instrument that we use to proxy an exposure must be very reliably and validly associated. And that's where genome-wide association comes back in again. And these are usually replicated in independent data sets, so we know that those instruments are reliable. And the effect must go via the exposure directly to the outcome. There should not be any effect going via an independent pathway to the outcome, and we also check for confounding with other measured or possibly unmeasured factors as well, using sensitivity analyses. And there are further extensions of the method that have been developed more recently, such as multivariable Mendelian randomization, where we can decompose direct and indirect effects of very closely correlated risk factors, which is of interest to us. For example, here with smoking and alcohol, on a single disease outcome, such as head and neck cancer. So just getting into the results then. This paper aimed to actually do that, which was to unpick the independent effects of both smoking and alcohol, which has been traditionally quite difficult to do using observational epidemiology. And very often, most of the papers found that alcohol really was only effective synergistically with smoking. But here, as you can see, not very clearly, but in the bottom, there is an independent effect of both smoking when accounting for alcohol and alcohol when accounting for smoking, particularly in the multivariable MR models. And these odds risks are increasing the risk of both oral and oropharyngeal cancer at around about the same effect estimates. It must be said that it's difficult to directly compare observational epi with MR, mainly given the differences in methodology and interpretation, particularly because with MR, you're proxying a lifetime exposure, whereas with observational epidemiology, you might be capturing a window of time in the exposure. We also want to do some further individual level analyses to look at how smoking could be actually modifying the risk of alcohol here. But some interesting findings from this paper. We took the technique a step further and looked at the genetic proxied effect of sexual behaviors. So you can see here on the top left, with later age at first sex, there was actually a protective effect found in oropharyngeal cancer risk, and with increased number of sexual partners on the right, an increased risk of oropharyngeal cancer, which we would expect. But just to demonstrate, using the multivariable model, when we accounted for the other sexual behavior, as well as alcohol and smoking and educational attainment, these effects actually attenuated more towards the null and completely crossed the null in the case of number of sexual partners. We also carried out a number of sensitivity analyses, including this cause method, and actually identified correlated pleiotropy, which is a major problem when you're trying to use genetic proxies, because some of these variants which proxy for sexual behavior might also increase your risk of drinking alcohol or smoking that could be actually leading to the outcome of head and neck cancer. And as part of this work, we conducted a novel genome-wide association study using the 10,000 participants with serology samples in UK Biobank. And you can see that there's a relatively low seroprevalence in Biobank, under 1% for most of these markers. And as we've discussed earlier in the week with Tim, we probably need to think about using the other 10,000 samples from Epic Norfolk, if that's possible, or waiting for the 50,000 samples to be done in UK Biobank, which should be around 2024, we think. We did manage to perform some LD score regression, but got very weak correlations between sexual behavior and the HPV seropositivity markers, given that it's pretty underpowered at the moment. And then moving on to some of the less well-established risk factors for head and neck cancer, as you can see in the forest plot, there's relatively limited effects using Mendelian randomization when we genetically proxied adiposity measures such as body mass index, waist circumference, waist-hip ratio, range of glycemic traits, including type 2 diabetes and hypertensive traits as well, with results generally sitting in around the null. This may suggest that observational studies, many of which show protective effects and risk effects for these metabolic traits, could be confounded, maybe overestimated. There could be reverse causality. But at the moment, as Paul mentioned during the week, the genome-wide study that we have for head and neck cancer outcome is probably relatively underpowered for this analysis as well. And we need to look at the new GWAS that Tom presented going forward to replicate some of the findings. We wanted to unpick this relationship a little bit further to determine why observational studies might be finding associations between metabolic traits and head and neck cancer. So we performed MR using metabolic traits onto known risk factors, and we did find some effects for smoking and alcohol in particular, which could suggest mediation of the relationship. And then finally, some of the work I was interested in was using genetic proxies for drug targets, and this has been quite well established in cardiovascular disease, Mendelian randomization studies, whereby they've identified SNPs within genes such as HMG-CoA reductase to act as a statin instrument, and others within ezetimibe, CETP, and PCSK9 inhibitors. And if this were cardiovascular disease as an outcome, you would see very strong protective effects with these instruments. But as you can see here, most of the results line up along the null, limited evidence for an effect on oral or oropharyngeal cancer risk with these drug target proxies. And it could be that, again, there's issues with the observational studies that are coming out suggesting statins are protective for head and neck cancer. We did, however, get a surprising finding with PCSK9, whereby when we inhibited this more novel cholesterol-lowering therapy using drug proxies, we found an increased risk, particularly within the oropharynx, which did replicate in UK Biobank. There was relatively good concordance between the two studies here. So I followed that up as an exploratory chapter in my thesis. The gene itself actually works by inhibiting LDL cholesterol receptor recycling. So when you actually block PCSK9, you get more receptor recycling to the cell surface that internalizes cholesterol, and therefore you get lower blood cholesterol, which is why it's of interest in cardiovascular disease. But we hypothesized this process could be pro-tumorigenic in oropharyngeal cancer, and also there was relatively good evidence in the literature that PCSK9 is also involved in regulating CD81 tetraspanins, allowing hepatitis viral entry in hepatocellular carcinoma. So given that their effect was in the oropharynx, we secondly hypothesized it could play a role in HPV entry as well. I used the GWAS that we had from UK Biobank in an almost kind of two-step fashion to first look at the effect of PCSK9 inhibition on HPV markers to see if that determined an increased risk of HPV seropositivity. And there was, albeit wide confidence intervals, but an increased risk of HPV 16E6, which is more known to be associated with sexual transmission. But when we followed up with some experimental work with collaborators at the University of Münster, where we infected keratinocytes with HPV 16 pseudoviruses, we didn't find any difference in the number of pseudoviruses virions delivered into the keratinocyte cell nucleus when PCSK9 was knocked down versus the scrambled control. So we haven't yet found a clear role with that hypothesis. So just to wrap up, there looks to be an independent effect of alcohol when using Mendelian randomization on oral and oropharyngeal cancer risk. And this could help really explain the sort of more recent changes in etiology with the decline in smoking rates. MR doesn't appear to be an appropriate method in this case when proxying sexual behavior, and there's probably caution to be used when proxying complex behavioral traits, given the potential for correlated pleiotropy, and therefore you need to ensure robust sensitivity analyses, possible multivariate approaches, and triangulation with observational and experimental evidence where possible. There was limited evidence for the effect of metabolic traits, but again, these probably need to be replicated in the new GWAS, and some plans to do further characterization of PCSK9, which is currently undescribed in oropharyngeal cancer, and I've got some plans for that if anyone wants to talk to me about it after. Thank you.
Good morning to everyone. I am Paolo Vasapolli. I work at the Cancer Epidemiology Unit of the University of Turin, and I'm going to present our project entitled The Role of Line 1 DNA Methylation. As you know, head and neck squamous cell carcinoma is a multifactorial disease which is caused by many different types of agents. For example, the patient's genetics, their lifestyle or habits, or could be also caused by environmental factors and viral infection. In literature, there are many different papers and articles that described and confirm that head and neck cancer could be related to some specific alteration in genes, alteration in genes expression, and also in genes sequence. One of these alterations is gene methylation, which has been observed as a reliable biomarker of tumor diagnosis and prognosis. The line 1 gene has been demonstrated to be a very good proxy of global methylation and a promising biomarker of prognosis in many different types of tissues, including head and neck cancer. And for this reason, we decided to deepen our interest into this gene because the line 1 methylation could be a biomarker, a very good biomarker for head and neck cancer. The purpose of our study was firstly to perform a systematic review of primary studies, evaluating the prognostic role of line 1 DNA methylation in patients with head and neck. Then we wanted to evaluate line 1 methylation levels in different types of biological samples of the same patients, starting from tumor tissue, then moving to non-tumor adjacent tissue, the margin, the buffy coat, plasma, and saliva. This will permit us to understand if line 1 could be used as a prognostic biomarker, and then also if there are some easily accessible biological samples which could be used as a surrogate for a global methylation status in the tumor tissue. So the systematic review starts from the National Library of Medicine database, in which we search for some specific, using some specific keywords in order to select only the papers related to some specific cancer. Here you can see the subgroups of cancer that we were looking for. Then we also use a subgroup of keywords related to the line 1 gene and the gene modification, hypomethylation and methylation, and also use a specific subgroup for the prognostic outcome that we could have from this type of studies. This literature was also filtered using an inclusion criteria. For example, for the population, we only selected papers with all those diagnosed with head and neck cancer without discriminating gender, ethnicity, implicated etiology, TNM staging, or histologic grading. And also we were looking for some specific head and neck subsite studies, in particular the one related to the oropharynx, oral cavity, pharynx, and larynx. We obtained 313 papers, which were firstly screened for their titles, discarding 220 of them, and then the 93 remaining were screened for their abstract, and 83 of them were excluded. The remaining 10 papers were analyzed in order to extract the most important information related to the studies, such as the subtype of cancer that they used in their studies, the number of patients used in their populations, which varied from 25 to 346, then the type of samples used for the analysis, and the techniques used for the DNA methylation analysis. Another important result that we were looking for is the prognostic outcome related to the line 1 DNA methylation. What we observed is that in 5 out of 10 of these studies, there was a result related to the prognostic outcome. In particular, all 5 of them found an association between line 1 hypomethylation and a worse prognostic outcome, in particular with disease-free survival, relapse, and a disease stage which was used as a proxy for prognosis. One example is the study which was done in 2017, in which they used a population of 77 oropharyngeal carcinomas. And you can see by their data that with a decrease of 10% of line 1 methylation, they had a result of an increase of 50% of risk of relapse. Then for the DNA methylation analysis, we used a population of patients which were recruited for the Healthspace Turing project with at least 2 biological samples available independently of the tumor subsite. While for the DNA methylation analysis, we've performed a pyrosequencing. In our preliminary data, what we observed with a population of 21 patients is that the methylation level in tumor tissue is way lower compared to the other different tissue, the non-tumor adjacent tissue, the margin, and the saliva. And what is also interesting is that by moving away from the tumor site, there is also an increase of the methylation level, suggesting that there might be a field sensitization effect in this kind of tumors. Then moreover, we also try to understand if there is a correlation between these different type of tissues. And what we observed is that between the tumor tissue and the non-tumor tissue, there is a very strong correlation, which was also seen in with less strength between the margin and the non-tumor tissue. While with the saliva, we observe that there is no correlation among the other 3 different tissues. Then the final analysis that we have done is a stratification analysis based on the tumor subsite. And as you can see by the data here related to the tumor tissue, there is all the methylation is very dependent on the tumor subsite. Where you have the oral cavity cancer, you can see that there is a higher level of methylation compared to the oropharyngeal one or the laryngeal one. This trend is also kept and confirmed in the non-tumor tissue, where unfortunately we did not have the oropharyngeal tissue, but is still kept between the oral and the laryngeal one. This trend was not conserved and was not observed in the saliva, as you can see by the data, and so we did not see any kind of trend in this case. So to conclude, what we were able to observe by this analysis is that from the systematic review, we observed that there are only few papers that study the correlation between line 1 methylation and tumor progression, but all of them found an inverse association. From our preliminary data, we were able to observe that there might be a field cancerization effect in line 1 hypomethylation, but no evidence was shown of correlation of line 1 methylation levels between saliva and tumor tissues. Since these are only preliminary data, what we are going to do is to firstly analyze also the buffy coat and the serum samples of the patients. We're next going to increase the number of patients in order to improve our data. We are going also to focus on tumor progression, and would be also interesting to compare the HPV positive patients with the HPV negative one. I want to thank all the units that are taking part of this project from the University of Turin, and in particular the Cancer Epidemiology Unit with Lorenzo Ricciardi, which is the principal investigator, and my lab mates, Valentina, Maya, and Giorgio. And I want to thank you all for your attention.